Okay, good morning, everybody. I think we can get started. Welcome everybody to the NEXUS User Day and also the NEXUS User Assembly. My name is Niko Beerenwinkel. I'm currently the chair of the steering board of NEXUS. And I'm very happy that all of you made it into this virtual meeting this year. I think we have a very interesting and very exciting program. And I would first like to briefly share the program for today with you and explain roughly what we're going to do. So in this first 15 minutes or so, I would just say a few words, and then Daniel and Christian will share with you a few updates about NEXUS. And then throughout the morning, we have a series of, I think, very exciting presentations that are usually done by clients of NEXUS, so people who have run specific research projects, and from people from NEXUS who have supported these projects. And I think it's a very nice variety of projects that we will hear about. There should be a coffee break around 10:30, and then the morning session runs until 12:15. There will be a lunch break until 1 p.m. And then we have the user assembly at 1 p.m. It usually doesn't take very long, but we have definitely blocked for it 45 minutes. And then after that, there is a keynote presentation by Lodewijk Wessels from the Netherlands Cancer Institute, and he will talk about drug response prediction. So that's the program for today. You will see a number of projects and to some extent their results. And then we have our user assembly, where the main task that we have is to vote on the new steering board for the period 2021 to 2023. If you can go to the next slide, Daniel, thanks. Well, I just very briefly want to remind you that NEXUS is in so-called ETH technology platform. This means that it is organizationally part of the Vice President Research of ETH Zurich. Well, there's a steering board, and then we have two units in this technology platform, the so-called Aerognostic Discovery Unit, or TDU for short, which is run by Christian Stirnimann, and the Clinical Bioinformatics Unit, or CBU for short, which is run by Daniel Steckhofen. And most of you have probably interacted with one, and most of you with only one of the two units, but we will see a number of examples from both units today. So as I said, Daniel and Christian will share a few updates about it with you. I have only one piece of news that I want to share before I give the word to the two of them. And that is that NEXUS also has a scientific advisory board. And these are the people on the advisory board. So we are very happy that they agreed to serve on the board. It's Lodewijk Wessels from the Netherlands Cancer Institute. I mentioned him already because he will give the keynote presentation later. Then we have Sonja Sievers from the Max Planck Institute for Molecular Physiology in Dortmund in Germany. We have Claudio Thoma. He's a senior principal scientist at Novartis, actually working in Cambridge in the US. He's also an alumni of ETH Zurich. And we have Michael Berthold. He's also a professor in Germany in Konstanz, but is currently, let's say, mainly and mostly the chief executive officer of a company called KNIME. So we're very happy that these four people agreed to serve on the board. And in fact, right after the user day today and the user assembly in the afternoon, in the early afternoon, we will have the scientific advisory board meeting together with these four. And they will also be present, at least in part, during the day-to-day. So if they are around and ask peculiar questions, then you know why. Okay, with that, I hope we're going to have a very exciting and interesting day. Thank you, everyone, for joining already. And please, Daniel and Christian, you can take over.
Thank you very much, Niko. As Niko told you, NEXUS is an ETH technology platform. So the goal is to provide services to research groups inside and outside of ETH Zurich. Outside of ETH Zurich, that is because we are an outreach and innovation platform enabling biomedical research and development. For one part, basically bringing fundamental research tools and technologies into said projects, but also to basically involve ETH Zurich in the biomedical research landscape. So my group, the Clinical Bioinformatics Unit, has three main divisions, being bioinformatics, where we basically are able to help you with the sequencing, which is done at another platform, but take raw data and put them into context. We have lately developed quite some experience with single-cell genomics and the afterwards cell typing. You will hear more about this in the talk later in the morning. And we have also made tremendous experience with research data management systems as part of the tumor profiler study that I assume many of you know. The second leg is statistics. So we help you with experimental design, statistical evaluation, such as inference, deep learning, modeling in general, up to the point of a publication. So this means visualization, writing the right text for the methods part, but also helping you with your ethical applications. The third and newest leg that CPU has established is software engineering. So software engineering, for one part, it has to do with the whole interoperability idea of initiatives like SPHN and PHRT that try to enable seamless transition between routine data, research data, even citizen-tracked data. So databasing and API development has become a daily bread and butter for our unit as well. But it does not end there, because in the end, we also want to report back derived results to sometimes non-technical end users. So front-end development has become a thing too. The newest embarkment is into the development of mobile apps, enabling user or patient reported outcome data, lifestyle information back into research projects. And now I hand over to Christian.
Thank you very much, Daniel. So as already mentioned by Niko, I'm leading this Aerognostic Discovery Unit, which also has kind of three pillars we work on. One of our major tasks is to support the research groups in high throughput screening. There we don't just do state-of-the-art screening, so we also try to go with the time and offer really cutting-edge technology such as organoid screening. So we've done various screens with organoids in the last 2 years. But on the side, we also offer classical 2D cellular screens, but also biochemical assays that we automate and do high throughput screening with. As we also will hear later, we are also working now on miniaturization of these assays to reduce sample volumes and so on. The second pillar we are working on is general lab automation technologies. We offer tailored methods for the need of the researchers at ETH. So we spread the field now also towards more chemical based projects. So we have currently one project running with a group that is involved in chemical synthesis. And we also can offer standard methods for DNA purification or bacterial library management. We've also done yeast library management. That's roughly on that side. Then on the user lab side, we are currently extending the user lab. We have started with offering a few readout devices, which was a Tecan M1000 plate reader, as well as an Acumen for high content imaging, so which is a fast device to collect images from cells with fluorescence label in. But now we have acquired an OpenTrons robotic system. That's a simple, small liquid handling system with which people can by themselves run smaller assays by themselves, and they get trained by us and can then use the device upon booking. With that, probably go to the next slide, Daniel.
Yes, thank you, Christian. So here we just list a couple of milestone Lighthouse projects that not only put NEXUS in some sense on the map for being a strong partner, but some of these projects also enabled us to learn new tools and technologies and implement them into our portfolio. I mean, I assume that most of you know the tumor profiler study, and we will hear a lot about COVMAS today. We have also been collaborating with a medical institute in Spain to build a cloud-based genomics pipeline and a GUI, a front end to allow them to interpret variants in rare disease. On the right-hand side, you can see successful proposals with large funding bodies, such as a small molecule high throughput screen with the SNF. This is actually a Synergia project. Some of you may know the Swiss Variant Interpretation Platform, which is an SPHM project, and the Swiss Molecular Pathology Project, where we've been building one of these interoperable CBio portals, which is also secure, so we're proud of that. But we're not only doing big projects. Here you can see many other successful projects, which are more like they have more like a service character. So these projects are smaller, have clear goals, and NEXUS provided parts of it to our customers. In the interest of time, I will not go through this list. You will be able to receive these slides and also the cost of the webinar later on our YouTube channel. Back to you, Christian.
Yeah, so one of the focus areas on the lab automation side and screening side was that we realized that ETH has a broad variety of different technology that can be used for screening available. And these different areas are organized in different platforms of ETH. So we have ScopeM, which is very strong in high content screening and the analysis. We are strong in high throughput screening in general and lab automation, but we also have a flow cytometry core facility at ETH, which can do cell sorting that also can be used as a readout for screening. We have the biomolecular NMR spectroscopy platform that can support screening projects using, for example, screening with smaller fragment compounds. Then we have a lab automation facility at the DBSSC in Basel that also automates workflows, also automated cell culturing and so on are their strengths. And the newest platform available is Gamel, which is very strong in genome editing. And having all these 6 platforms together, we decided to found a meta platform that can offer tailored screening campaigns to the ETH researchers, and really design a project for these researchers on their individual needs. It's called Screening at ETH, so please also visit our website there. And we are currently building up that infrastructure and hoping to be able to run a lot of projects through that infrastructure in future then.
Of course, all these great things are possible only with a great team. So this is the current staffing of NEXUS. We're a lean but mean platform, and we're looking forward to continuously collaborate with you guys. And if you have not been working with us, feel free to contact us through any means possible. And we are happy to help you out, get your biomedical research going and accelerated.